Hey everybody, happy Wednesday. I'm coming to you again with another recorded lecture. Before I start, I wanted you guys to know that I'm hoping to come to back to school night tonight if my doctor sends me my off work release. I cannot come until I get a note from the doctor that I'm allowed to come back to work. So if I am going to be able to make it, I'll send out a remind and let you guys all know. But I'm hoping to come tonight, and then I'm really hoping to be there with you guys Thursday and Friday and so on. So if you haven't already joined our remind, make sure that you do join it. All right, the next topic that we're going to move into are enzymes. And enzymes help chemical reactions happen faster without getting used up. They're like the matchmakers of the cell. Enzymes typically are protein molecules. And remember our 4 groups of organic compounds? We had carbohydrates, right? Lipids, which are fats, oils, and waxes. Nucleic acids. And proteins. So enzymes are a subcategory of proteins, very important for helping cells run all of their chemical reactions and get things done in a timely enough fashion that it makes it possible for a cell to survive. First of all, what is a chemical reaction? A chemical reaction is a process by which elements combine and interact with each other and you make new things. For example, element A plus element B, they chemically react with each other, which means they're going to make either an ionic bond or a covalent bond, and they give you element called C, which is different in its properties from A and B. For example, sodium and chlorine gives you sodium chloride. Okay? Everything here on the left side of the equation, these are called the reactants. They're reactants because they react with each other. And everything on the right side over here, these are called products. This is what is being produced. Now notice on this side, I've got 1 sodium and 1 and I think that thing down there where it says Doceri desktop, I think that's in the way, so I'll write it over here. 1 sodium and 1 chlorine. And if you have one of each of these on the product side, then you better have 1 sodium and 1 chlorine over here for the equation to be balanced. You cannot create or destroy matter, so everything has to be balanced. If I look at another equation where I'm making hydrogen gas plus oxygen gas with a little bit of electricity gives us water. These are our reactants. This is our product. But is this equation balanced? Do we have the same number of elements, atoms on each side? So I've got, how many hydrogens? I've got 2. And how many oxygens over here? I've got 2. On this side, though, I have 2 hydrogens, see the 2 there, but how many oxygens? Oh, I only have 1. So how do I make it balanced? Well, because I need to have 2, if I add a 2 out here, this is called a coefficient. Now I've got 4 hydrogens and 2 oxygens. It's still not balanced because now I've got 4 over here. So how can I make it so that I've got 4? Well, if I put a 2 in front here, now I'll have 4, and now my equation is balanced. And so that's why sometimes you'll see these numbers out in front of compounds or molecules. So we can add these and change these, but we never mess around with the subscripts. Okay. So a chemical reaction is when things get rearranged. Atoms can get rearranged and give us new things. So sometimes we have something like, if we have it like this, AB plus CD, and you can rearrange things. You can have A go with D and B go with C. So that's a double displacement reaction, and you'll learn a lot more about this in chemistry. So sometimes these chemical reactions happen very, very slowly. reminding you back of the picture at the beginning of the lecture where we had a metal and iron chain that was being oxidized with oxygen and water in the presence of oxygen will oxidize iron, but it's a very slow process. And then sometimes if you're looking at something like an explosion, a chemical reaction can happen with great energy and it can happen very, very quickly. Think of a firecracker. Just to remind you, the stuff that's on the left side of the arrow, those are the reactants, and the things that are on the right side of the arrow are products. And so, here's your first key question. Remember, you're going to want to write this down and use it in your left-hand column on your notes. What happens to chemical bonds during chemical reactions? So, maybe you might be able to come up with that answer by looking at this, AB plus CD. Now, there's no A and B elements. We're just using those for samples, right? C, B, C. So if you have this molecule or compound called AB, it's got some type of chemical bond holding the A to the B, and the C has some type of chemical bond holding the C to the D. Well, what do you think is going to happen to these chemical bonds during a chemical reaction? Well, the chemical bonds have to be broken and then reformed, because now we have the chemical bond between the A and the D and the chemical bond between the B and the C. So you're going to be rearranging chemical bonds, breaking chemical bonds, and making new chemical bonds when you're doing chemical reactions. Okay, so chemical reactions always involve changes in the chemical bonds that join the atoms together, changing and rearranging. So you don't need to write down the whole sentence. You can just write down the stuff that's in the, it's highlighted. And if this is going too fast for you, you can pause and go back and listen to it again. Okay? But it's just rearranged. Remember, whatever we start out with, we have to finish with. Matter and energy cannot be created or destroyed. It can be just rearranged and transferred. So, energy of reactions. Whenever a chemical bond is broken, energy is released. And whenever a chemical bond is formed, energy is absorbed. Okay, so if you break chemical bonds, breaking chemical bonds, energy, capital E for energy, is released. And if you make bonds, energy is needed. So keeping in mind the big picture, the food that we eat, the food that we eat goes into our gut where bonds are broken, And so energy gets released from that process. That energy then is used to form new compounds. New molecules. In order to make these new molecules and compounds, you need energy. See, in order to make the bonds, you need the energy that was released from breaking down things. So we're constantly just like this recycling plant where we're taking things in a form that we don't necessarily need and turning it into something that we do need. And that's what digestion and metabolism is all about. Oops, jumped too far, sorry. So how do energy changes affect whether a chemical reaction will occur? How do we know if a chemical reaction is going to happen in the body? If energy is released, then the reaction is going to happen spontaneously, which means, so releasing energy is spontaneous. That means it happens by itself. But if energy needs to be absorbed, if it requires energy for it to happen, then it has to have that source of energy. So energy is needed. Okay, so what lets us know if a chemical reaction is going to happen? Well, if it's a chemical reaction that releases energy, it's going to happen all by itself. But if it's a chemical reaction that needs a little push to get it started, then energy is going to be required in order for it to happen. in order for it to occur. So when hydrogen gas, and we already looked at this equation already, when hydrogen gas reacts with oxygen to make water, this is an energy releasing reaction in which energy is given off as heat. And so, like I said before, you have to have those number twos out in front of the hydrogen and the water in order for the equation to be balanced. Okay. In order to break water molecules down and to give us hydrogen gas and oxygen, it requires a lot of energy in order for that to occur. So in order to stay alive, organisms need to carry out reactions that require energy. Just like I was talking about a minute ago, the food that we eat goes into our digestive system, and we're going to be breaking apart the large molecules, the large proteins and lipids and carbohydrates that we're eating in our diet, breaking them down into their smaller monomers, their building block molecules, and then using those building block molecules to give us the energy that we need for our cells to function in the way they need to. Remember, matter and energy are conserved, so we have to have a constant input of energy into our body. Think about the bead lab that you just finished up. You have to be constantly supplying new beads, new elements, so that you can break them apart and reassemble the molecules that you need in order to survive, whether they're amino acids or simple sugars or water molecules. If you don't have the raw materials that you need, can you have a healthy body? No, you can't. You can't make the things that you need in order to survive without having all of the raw materials that you need. That's why eating a healthy, balanced diet is so important. So plants get their energy from the sun. Remember that process, photosynthesis, should sound familiar, where plants take in sunlight and use the chloroplasts and the chlorophyll molecules, and they take in water vapor from the soil, and they use that in order to make sugar, glucose molecules. So where do we get our energy from? Okay, can we get it from the sun? No. We have to get our energy from eating plants or other animals directly. So in a roundabout way, we are all dependent upon the sun, but we just can't sit out in the sun and make energy that way. We need to eat the animals and eat the plants. Activation energy. Activation energy, to activate means to start something, right? So activation energy is the amount of energy that it takes in order to get a chemical reaction to occur. So the activation energy is like the jump start. Think of the activation energy that's needed on a roller coaster. That's the system that pulls you up the hill at the very beginning of the roller coaster. And once you get to the top of all roller coasters start out with a big incline, right? You go way, way up the hill. That's your activation energy. Because from there on, it's just using your momentum to carry you on through the rest of the ride. Activation energy is the amount of energy that is required in order to get a chemical reaction to start. Okay, if the products of the chemical reaction are at a higher energy level than the reactants, then this is going to be an energy absorbing reaction. It takes energy to get it started. If the products are at a lower energy level than the reactants, then it releases energy, and that's going to happen spontaneously. Okay, so you can see it takes less activation energy here, and so these are going to happen more easily or more spontaneously without any energy input at all. Enzymes are special chemicals that they make life possible, because sometimes chemical reactions are much too slow, or they have an activation energy that is too high. And so it would take so long for them to happen inside of these chemical reactions for them to happen inside of the body that you wouldn't have a good survival rate as an organism. So we need to have these chemical reactions happening more rapidly, and this is where enzymes come in. These enzymes, we refer to them as catalysts. So what is a catalyst? A catalyst is a substance that speeds up the rate of a chemical reaction. It makes the chemical reactions happen more quickly because they lower the activation energy, the amount of energy that is required to make the chemical reaction occur. So they're catalysts. They speed up chemical reactions by making it easier for them to occur. So the example I always like to use is, do you guys know what a matchmaker is? Back in the day, people would go to dating services if they were looking to find somebody to potentially settle down with and get married. They would pay someone to help find them a match. Nowadays, we have computers, right? So when you go to a matchmaker service, whether it's online or an actual person, I mean, if you're really wealthy, you don't go online. You use a matchmaker service because you want to find the appropriate match, right? But let's say that you've got, I don't know, Phil over here, happens to be the name of my husband, and Brenda over here, that's me. And we both used a matchmaking service called Match.com, and this is true story. See, Brenda worked so much, and she was a teacher, and she was always spending her time with kids. And Phil is an architect, and he was really busy working 6, sometimes 7 days a week. And they just didn't have time to go out and look for people. So we both went to this service, Match.com, and we put in what we were looking for and what we were matched with. And this matchmaker helped to make the connection between Phil and Brenda. And then from there, it was up to us to do the chemical reaction, right? And now, 16 years later, we're married and have 2 kids. But once we were matched up, we didn't need Match.com anymore. So the Match.com, the matchmaker, that acts like the catalyst. It helped the chemical reaction to get together and bring 2 things together. So going with our pretend elements, they're A plus B, it makes the new thing AB. But it doesn't stick around. Enzymes, however, can also break things down. Sometimes they bring things together, but sometimes they break things down. For example, if you had AB and you wanted it to be broken down into its individual parts, an enzyme can also be responsible for doing that. In this case, this one's a matchmaker. I think of an enzyme that would break things apart as a divorce lawyer. Right? It's kind of sad, but it's breaking apart what was together and making it 2 individual pieces. And once the divorce is final, the lawyer is out of the picture. So, why are enzymes important to living things? As I had said before, enzymes are important to living things because they help chemical reactions go at a fast enough rate to make life possible. They are typically proteins, and they act as biological catalysts. They help chemical reactions happen faster without getting used up. They lower activation energies, and I'll go back because I know you probably didn't get all of that. Okay, so they speed up chemical reactions, they lower activation energies, they are proteins, catalysts, speed up chemical reactions, lower activation energies, and they can be reused, okay? Or they will be reused, okay, not can be. And they're reused over and over. So they themselves do not become part of the chemical reaction and they don't get used up. Okay, so with a reaction that uses an enzyme, look at how much lower the activation energy is. Look at the red line. The red line, it happens much more easily. The activation energy without the enzyme is much greater, and so the chemical equation would take a lot longer. Think of a matchmaker. If you're at a dance and you see someone across the room that you think is cute and you'd like to ask them to dance, but you're really shy, you might eventually through the night bump into them by chance and then decide to dance with them. But it's going to take you all night long before you finally bump into them. Whereas with an enzyme, if I'm at the dance and I know that you like someone else, then I'm going to act like the catalyst and I'm going to go over and I'm going to grab you and I'm going to grab the other person. I'm going to say, hey, I know you guys like each other. Why don't you go dance? So that's me acting like a catalyst. I don't want to be out there on the dance floor with you. I'm just going to help you guys get together and dance. And so it's important to note that enzymes are very, very specific. They usually only do one type of chemical reaction. And so here is a drawing, a computer model of a catalase enzyme. And this shows you how catalase actually has a 3-dimensional shape, and its job is to specifically do one thing. catalase. It breaks down hydrogen peroxide into oxygen and water. Now let's hopefully my little link here will work, and it will take us to an animation here which tells us how catalase works.
Each of the 4 subunits of the enzyme catalase has a cavity with an iron-containing heme group at its center. Hydrogen peroxide, the substrate of catalase, enters a cavity. One oxygen atom binds to the iron in the heme group. The other oxygen is attracted to an amino acid, a histidine, that projects into the cavity. The bond between the 2 oxygen atoms is strained and breaks. Water forms when the remaining OH fragment pulls a hydrogen atom away from histidine. In a later reaction, another hydrogen peroxide enters the cavity and pulls oxygen away from iron. This forms water and oxygen gas. The enzyme has now been restored to its initial state and is ready to act again.
Okay, so that's a nice little animation that shows you. It goes into a lot more detail than you really need, but I think it's a nice animation. Notice that catalase ends in ASE. Enzymes typically have that ASE ending, and then the root of their name is going to refer to what they break down. So lipase breaks down lipids, protease breaks down proteins, and so on. So, in order for chemical reactions to occur, the reactants have to collide. But if there is not enough energy, then they aren't going to collide and no reaction is going to take place. That's what the activation energy is all about. So, when the enzyme meets its substrate, that is the thing that it's going to break down. And it's going to combine together into what's called the enzyme substrate complex. When it does that, the energy that's needed to start this chemical process is released, or it's lowered so that the chemical reaction can occur. So the substrate is the thing that gets broken down. So for our example of catalase, okay, catalase is the enzyme, and hydrogen peroxide, H2O2, is the substrate. So hydrogen peroxide binds with the catalase. And when the hydrogen peroxide is bound to the catalase, that becomes the enzyme substrate complex. This is an example of glucose and hexokinase, which is its enzyme that helps to break it down. So when glucose and ATP, which is energy, bind together to the hexokinase, that's your enzyme substrate. Now the three-dimensional shape of these molecules is very specific, and so is the shape of the enzyme. So we call this the lock and key model for an enzyme and a substrate. Once the enzyme and the substrates are bound together, then there is some type of three-dimensional shape change, which is going to convert the substrate, your reactant, into your product. Sometimes it might be taking 2 smaller subunits and putting them together, and sometimes it's taking a large thing and breaking it down into 2 small things. And once the product is created, it's released, and the enzyme goes back to its normal shape, and it's able to do this chemical reaction over and over and over again. Okay? So on this screen, you see, first of all, a 2-dimensional model. And you see there, here is our enzyme, okay, hexokinase, and it has a 3-dimensional shape. And in its shape is this area here called the active site. That's where the 2 substrates, glucose and ATP, connect together. They bind to the enzyme, and this makes the enzyme substrate complex. The substrates are then converted into products. You'll see that there is a hydrogen that is passed over. And now you get, a phosphate group that gets passed over. And you get your products here, glucose 6-phosphate and ADP. And it starts all over again. You have, again, your enzyme ready to go. So there are 2 animations here that I won't play right now, but you should go back and look at them because we're get 24 minutes on this lecture, so I don't want to take up too much more time. But you have access to this lecture, and I want you to go back and look at both of these animations on how enzymes work, enzyme substrate complexes, because you're going to need to draw this in your interactive notebook. How are enzyme activities regulated? What kind of things control how often or how quickly enzymes are going to react? My computer's lagging behind me. Come on. So enzymes are going to work best at specific pH values, not too acidic, not too basic. Enzymes will work best at specific temperatures. Those are the two parameters that are really, really important, and those are the two that we're going to look at when we do a virtual lab in the next couple of days. If you take the enzymes out of those ranges of pH and temperature, they're literally going to change shapes so much that the substrate can't fit in anymore. It's as if you take and you use a blowtorch on a key and you melt the shape of the key so that it won't fit in the lock anymore. That's exactly what's going to happen. Cells also have proteins that can help turn on and off enzymes. So sometimes if a chemical reaction needs to occur, the enzyme will be active, and then when the cell doesn't need it anymore, it shuts it off. So enzymes, really important. They help to regulate chemical pathways. They help the cell make materials that it needs. Enzymes help to release energy, and enzymes help with transferring information throughout the cell. So let's do this really quickly. The elements or compounds produced by a chemical reaction are known as, here the key is produced. So the answer is products. Products are produced. Chemical reactions always involve changes in energy. The factor that prevents many energy-releasing reactions from occurring at relatively low temperatures is called activation energy. And remember, pause if you want to take a second and answer these. All of the following statements, which of the following statements is true? All proteins are enzymes. All catalysts are enzymes. All enzymes are catalysts. All catalysts are proteins. This is hard, so take a second and read through all of your answer choices. All enzymes are catalysts. So there are some enzymes that are not proteins. And lastly, what happens to an enzyme after the reaction it catalyzes has taken place? The enzyme is destroyed and the cell must make another. The enzyme holds on to the product until another enzyme removes it. The enzyme is unchanged and ready to accept substrate molecules. The enzyme changes shape so it can accept a different kind of substrate. What do you think? Think of the matchmaker. After you go to a matchmaker and you find someone to go out on a date with, do you need the matchmaker anymore? Nope. So the enzyme is unchanged and ready to accept substrate molecules. All right, you guys. Hopefully I'll be there in the next couple of days. I'm planning on it. And so best of wishes, and maybe I'll see you at back to school night. Thanks a lot. Bye.